Hello, this is Allie and Michelle, and today we're gonna get you through the process of making the baby lab. Enjoy!
In the first phase of meiosis 1, inner phase, cells undergo a round of DNA replication, forming duplicate chromosomes. During meiosis 1, we separate the homologous chromosome pair into two gametes. We randomly pick the chromosomes by flipping the coin. The head side of the coin will be the dominant gene, while the recessive will be the tail. The action of flipping the coin is known as randomization. Whichever chromosome pair is chosen will go to a gamete, which is the dominant in this case. So the dominant gene will go to the A gamete, and the other one, which is the recessive, will go to the B gamete. We do the same thing for both sperm cells and egg cells, all 10 times. After that, we move on to meiosis 2. During meiosis 2, we separate sister chromatids into two gametes. We don't have to flip the coin this time because each chromosome is homozygous, so it doesn't matter where it goes. As a result, we will have 4 gametes at the end, 4 sperm cells and 4 egg cells. The same number of chromosomes came from each parent. There are 10 chromosomes found in each gamete owing to the fact that there were 20 chromosomes to begin with, and the number of chromosomes is reduced by half by the process of meiosis. By flipping the coin, we choose which gamete of the 4 egg sperm cell will be chosen to perform fertilization. The first baby will be chosen between gamete A and gamete B of both egg and sperm cell. The head side will be gamete A, and the tail will be gamete B. Now that we get head, we will choose gamete A from both sperm and egg cell. The second baby will be chosen between gamete 1 and gamete 2 for both egg and sperm. Chromosomes from sperm cell will go into the container that has a tail. And chromosomes from the egg cell will go to the container without a tail.
After putting the chromosomes into the cells, we perform fertilization. During fertilization, the sperm swims to the egg since it has a tail and transfers its chromosomes into the egg cell. The egg cell then becomes a zygote, turning from a haploid cell with 1 set of chromosomes into a diploid cell with 2 sets of chromosomes. The karyotypes of 2 babies are created when we arranged each chromosome in each egg cell with its homologous chromosome by looking for matching pairs of chromosomes. After the karyotype is finished, we can look at each homologous pair and see what the genotypes are. Each genotype has 2 letters. If a genotype has at least 1 uppercase letter, it resembles a dominant trait. If a genotype has 2 lowercase letters, it resembles a recessive trait. Speaking of letters, if the 2 letters in a genotype are both lowercase or uppercase, it is homozygous. On the other hand, if there's 1 uppercase and 1 lowercase letter in a genotype, it is heterozygous. We can identify chromosomal mutations by looking for missing, also known as monosomy, which is caused by deletion mutation, or extra chromosomes, also known as trisomy, when there's 1 extra chromosome, or polysomy, when there are 2 or more extra chromosomes of homologous chromosome pairs. These chromosomal mutations are caused by a process called non-disjunction. In this case, we didn't have any chromosomal mutations. We were given a chart that indicates what each genotype's phenotype is. Using the genotype that we found from the karyotype, we can find the phenotypes of each baby. There weren't any babies that have the same genotypes and phenotypes because during the process of fertilization, different genes from the sperm are passed on to the egg, which also have different genes. This is called gene shuffling. The end.
Goodbye, Dai. See you next time.